in light of how meiosis works. So remember, we're all diploid, so we all have 2 copies of every gene, one from mom and one from dad, but each of those genes we might carry different versions from mom and dad. We might have different alleles. So let's take this yellow fever mosquito and let's just pretend, I don't know if this is true or not, but let's just pretend that there are 2 different alleles for wing length. You can have, a mosquito can have short wings or it can have long wings. And this particular mosquito was given different alleles by each of its parent. It was given a gene for long wings from its mom and a gene for short wings from its dad. And I've written these up here so you can see which is dominant, which is recessive. So we're going to track the short wing gene in pink and the long wing gene in red. And here I've given these letters so you can see which is dominant, which is recessive. So long wings is dominant to short wings. So it's indicated by the capital L. So long wings is dominant to short wings. So if this individual, if this mosquito has a long wing gene from dad and a short wing gene from mom, what is it? Heterozygous. So this is its, I might make this in blacks, easier. So heterozygous. It has 2 different alleles at this locus, so it's heterozygous. All right, and you might notice that each of these sister chromatids has the same, and they always will, because sister chromatids are clonal copies of each other. Remember, in S phase, so in G1, this cell did not have duplicated chromosomes. In G1, there was just 1 line for each chromosome, so it would have had really just 1 red and 1 pink, or 1 long wing allele and 1 short wing allele. But then in S phase, each of these chromosomes was duplicated. And the 2 duplicates are still held together at the centromere, meaning they're sister chromatids, but they are exact copies. So this chromosome was copied. So now you have 2 sister chromatids that are identical. So that's why they both have the long wing allele, and then these 2 sister chromatids are identical, so they both have the short wing allele. So how do these get redistributed to sex cells, and how do we then put that in a Punnett square? So let's see, let's draw. So in meiosis 1, the homologous pairs get separated, and we also have independent assortment happening. And I'm even going to do a crossing over event here. So at the end of meiosis 1, you have each of these daughter cells is going to have 3 duplicated chromosomes. Each will have 1 of each. But I'm going to provide a crossing over event on the gene of interest just to show that that doesn't affect the final results here. So this is a little different than the crossing over event I showed before. The crossing over event I showed before was to the squiggly one. I'm not crossing over the squiggly one now. So I'm showing independent assortment. Maternal and paternal chromosomes don't always go to the same daughter cells. Okay. And now let's put the alleles of interest. So we're tracking, oh, can I get the same color again? Oh yeah, okay. Here, this. Okay, so the black maternal is carrying that dominant allele for long wings, and blue is carrying the paternal chromosomes for carrying the recessive allele. All right, and then meiosis 2, let's split those up. I'm drawing it, I could draw it out and then show you a picture, but I think that it is helpful to see exactly how this is going to play out. So we're going to divide these sister chromatids in half for the resulting daughter cells. Oh, yeah, this here. And this one will be the one that has some blue in it. And this one gets separated for these 2. So remember, this one is dividing those sister chromatids down the middle and distributing to those two daughter cells. This one's dividing these sister chromatids down the middle, distributing to those daughter cells. Let's get some blue here. So this part goes here. Oops, I forgot. Here. Squiggly. Put a little bit of black in this one, right? Because that's where the crossing over event happened, that long one. Okay. Now, let's red. Okay. Maternal set had the long wing allele here, and the short wing recessive allele here. So it's always on the big chromosome, but the paternal sets indicated by blue are carrying the recessive. Remember, blue is carrying recessive because that's the paternal chromosome. Father provided recessive, mother provided dominant. So the black, always keep the red. So now you can see that every daughter cell has one copy. So this parent was heterozygous, capital L, lowercase l. Each of these sex cells, this is important for the homework, right? Each of these sex cells has only one. So this sex cell has the dominant, this sex cell has the recessive, this sex cell has the recessive, and this sex cell has the dominant. So what percentage of sex cells, if this was a male, what percentage of the sperm is carrying the recessive allele? 50%, 2 out of 4. What percentage of his sperm is carrying the dominant allele? 50%, right? So 2 out of 4. So this is what's important to figuring out, to drawing a Punnett square. So let's look at how a Punnett square then would be filled out from this. Oh, and so I'm going to fill out a Punnett square where both, so let's say that this is a father, but let's say this is a male. So this is a male mosquito, and he inherited a long wing allele from his mother and a short wing allele from his father. Let's say this male mosquito mates with a female mosquito of the same genotype, a heterozygous female, where this basically happened the same way. So let's see what happens then. What we showed in the last slide was a father that was a male mosquito that was heterozygous for wing length, dominant was long wings, recessive was short wings. Let's cross him with a female who has the same genotype. So I just showed you how a heterozygous individual, 50% of their gametes, 50% of their sex cells will receive dominant allele. So what you do with a Punnett square is you put the possible sex cells along the top. So we put one parent along the top. So 50% of the father's gametes will have the dominant allele and 50% will have the recessive allele, which is what I just showed you. And that same process, meiotic process, will happen for the mother. So the mother's gametes, 50% of them will have the dominant allele and 50% will have the recessive allele. So now let's fill this in. So if we cross this male and this female mosquito, both who are heterozygous, what are the possible genotypes and phenotypes of their offspring? Well, it's all up to chance, really, because it just depends which alleles from the dad mix with which alleles from the mom. It just depends on which sperm meets which egg. So we can't know for sure, but we can figure out the probability. So 50% of the male's sperm is dominant, long wings. So what if that meets that? So some of them will meet up with that 50% of the female's eggs that are also dominant. 50% of them will meet up with the female's eggs that are recessive. 50% of the father's short wing alleles will meet up with the female's long wing alleles, and 50% of the father's short wing alleles will meet up with the mother's short wing alleles. So you basically just fill these down and fill across. And so now you can see that 75% of the offspring will have long wings because long wings is dominant, long wings masks recessive. So these heterozygotes will have long wings, and this will have long wings. So anytime you sing a single, anytime there's any dominant allele present, that's the one that's expressed. So you have 25% are homozygous recessive, 25% are homozygous dominant, and 50% are heterozygotes. But this one received the dominant allele from mom, this one received the dominant allele from dad, but still, so when you put them together, 50% are heterozygotes. But that means 75% express the long wing allele. So when you look at them, you'll see that 50% have long wings, even though 50% of them are heterozygotes, or carriers for the short wing.